Let's talk Streptococcus. I know, when you're prepping for exams, this topic can feel like a total beast, but we're gonna crack the code together. I'm gonna show you how to break this all down in a way that's super logical and, most importantly, easy to remember. Okay, so here it is, the whole shebang. And yeah, I get it, looking at this chart for the first time, it's a lot. Kind of intimidating, right? But seriously, stick with me. By the end of this whole thing is gonna make perfect sense. It'll be your go-to roadmap, I promise. First things first, let's nail down the absolute basics. This is non-negotiable for your exam. So Streptococcus, they're gram-positive cocci and they hang out in pairs or chains. But here's the key differentiator from staph, and you have to know this. They are catalase negative. Got it? Gram-positive cocci and chains, catalase negative. Lock that in. Okay, so when we start classifying them, the very first split on our roadmap is actually super simple. It's all about one thing, how they feel about oxygen. So this one question splits the whole family into two big groups. On one side, you've got the aerobes and facultative anaerobes. These guys are fine with oxygen. On the other side, you've got the obligate anaerobes, like Streptococcus. For them, oxygen is literally poison. Now, for our purposes, we're going to follow that first path, the ones that can handle oxygen. All right, step 2. And this one is huge. I'm talking classic exam question material, so definitely pay attention here. It's all about what they do when you grow them on blood agar. What we're looking for is something called hemolysis. And the name tells you exactly what it is, right? Hemo for blood and lysis for breaking down. So it's literally the bacteria breaking down the red blood cells in the agar. This gives us this amazing visual clue that helps us figure out who we're dealing with. And this breaks down into 3 types you absolutely have to know. First up is alpha hemolysis. Think A for almost. It's partial lysis, and you'll see this cool greenish color. Then you have beta hemolysis. Think B for big or bad lysis. It's complete destruction, leaving a big clear zone. And finally, gamma, which, fun fact, isn't really hemolysis at all. It means G4 goes nowhere. No change. Easy, right? Okay, let's keep going down the flowchart. We're going to focus on that beta hemolytic group, the ones that cause complete lysis. Why? Because this branch is where a ton of the really important human pathogens hang out, including the famous Streptococcus pyogenes. So how do we tell all these different beta hemolytic guys apart? Well, for that, we have to thank Rebecca Lancefield. Way back in 1933, she developed this amazing system called Lancefield grouping. It's all based on serology, basically looking at specific antigens on the bacteria's surface. And here is a golden nugget for your exams. Seriously, you're going to love this. Just remember the 4 C's. Lancefield classification is based on the C carbohydrate antigen in the cell wall. 4 C's. How easy is that? There's no way you're going to forget that now. So, using this system, Lancefield was able to split all the beta hemolytic strep into 20 different serological groups. And these groups are identified by letters, from A all the way to V. But, and listen up, because this is a classic trick question on exams, they skip the letters I and J. So no group I, no group J. Don't get caught on that one. Now, out of all those 20-some groups, here's the one you really need to focus on. Group A. Why? Because the vast majority of human strep infections are caused by group A Streptococcus, better known to all of us as Streptococcus pyogenes. But you know, in microbiology, we can always get more specific, right? Especially within group A. For things like tracking outbreaks, we need to drill down even further, and there are 2 more layers of classification you should know. First up is something called Griffith typing. And guess what? I've got another killer mnemonic for you. Just think good morning. The G from Griffith typing is based on the M from M protein. Simple. It's another one of those key antigens sitting on the cell wall. And this level of detail is just incredible. Using the M protein, we can subdivide group A strep into more than 150 different serotypes. That's a huge amount of variation. And finally, we can get right down to the molecular level, basically to the bacteria's source code. We do this with genotyping. We're looking at the M gene, which is the specific gene that actually codes for that M protein we just talked about. And if you thought 150 serotypes was a lot, genotyping blows that away. It reveals an even greater diversity. We now know of over 200 different M genotypes. I mean, the level of detail we can get to is just mind-blowing, isn't it? And there we are, back to the big picture. Take another look at this chart now. It's not so bad, is it? We just walked through the entire thing, step by step, from that simple oxygen question all the way down to the nitty-gritty of their genetics. So let's recap the key takeaway. It's just a logical 4-step process. Step 1, oxygen needs. Step 2, hemolysis on blood agar, alpha, beta, or gamma. Step 3, if it's beta hemolytic, use Lancefield grouping. And step 4, if it's group A, you can drill down even further with Griffith typing and genotyping. See? It's a really elegant, organized system when you break it down. And look, this whole framework isn't just about passing an exam. It's absolutely vital in the real world, in the clinic. Which leaves us with a pretty big question to chew on, right? If there are over 200 genotypes of just group A strep, how does that incredible diversity affect how we develop treatments and maybe, just maybe, a vaccine?